Share screen. Okay. Let's see. More. Hi, my name is Karla Perez. I am from section. Why can't you see it? Hold on. Control. Escape. Um, Hi, my name is Karla Perez. I am from Section 2, and today I will be presenting a research article titled Conditioned Variation in Heart Rate During Static Breath Holds in the Bottlenose Dolphin, also known as Tursiops truncatus. This was published in 2020 in Frontiers in Physiology by Fallman and colleagues. This study was also conducted at Dolphin Quest in Hawaii. My name is Karla again, and let's get started. So for some background and physiology, bottlenose dolphins, like all marine mammals, perform long breath holds while diving. When they do this, they rely on something called the mammalian dive response. This includes a pronounced drop in heart rate, known as bradycardia, the slowing of heart rate, and peripheral vasoconstriction to conserve oxygen for vital organs when they dive. This physiologically important This is physiologically important because it helps them extend dive time and manage limited oxygen stores underwater. They can't always come up for oxygen, so they do need to manage these stores when they are diving. And this ties directly into concepts we learned in class about the autonomic nervous system control, cardiovascular physiology, and the mammalian diving reflex, which I learned a lot about researching this topic. Like we discussed, the parasympathetic system slows heart rate, and this paper explores how that response might be modified or even controlled by learning. So for question and hypothesis, the main question the researchers asked was, can bottlenose dolphins voluntarily adjust or condition their heart rate response based on the type of breath hold they expect to perform? The hypothesis was that dolphins would show different heart rate patterns depending on whether they were preparing for a short breath versus a long breath, suggesting a learned or anticipatory component to the dive response. This is significant because the dive response is typically considered reflexive. If dolphins can anticipate and change their physiology ahead of time, it means that marine mammals have more cognitive control over cardiac function than previously thought. So it helps determine how flexible mammalian physiological responses can be. For my method summary, in this study, the researchers worked with trained bottlenose dolphins, Tursiops truncatus. Each dolphin was instructed to perform two breath hold tasks on cue. Short breath holds lasting around 30 seconds and long breath holds lasting over 2 minutes or 120 seconds. While the dolphins performed these tasks, the researchers recorded instantaneous heart rate using ECG suction cup electrodes on these dolphins. They analyzed heart rate right at the start of the apnea and throughout the breath hold. They used a mixed effects statistical model to compare the heart rate responses between short and long trials. This allowed them to determine if the differences were significant while accounting for repeated measurements from the same animals. So for um, figure one shows the instantaneous heart rate patterns for each dolphin during the long, short, and no symbol breath holds. So it would be this A panel right there. Each line represents the mean heart rate over time, averaged across all dolphins. Um, and for panel B, which is this one right here, C right here, and D right here, those are the three dolphins that were trialed during this research. What we can see is that dolphins preparing for long breath hold begin the trial with a much sharper drop in heart rate, a rapid, strong bradycardia. Short trials show a slower and more mild decrease in heart rate. You can kind of see that right here. And statistical analysis using mixed effect models found these differences to be significant with p-values less than 0.05. So it was significant. Physiologically, this means dolphins are adjusting parasympathetic activation depending on the kind of breath hold they're expecting to perform. In other words, they anticipate oxygen demand and modulate their heart rate ahead of time. As you can see, each trial, like I said, is each different dolphin. I just included figure one again on the next slide in case it wasn't visible, but didn't end up really using it. The baseline was -20 to 0 and up to 230 seconds or 40 seconds during a breath hold for a dolphin. So dolphin B, C, and D. For my results for figure two, this figure provides individual examples of heart rate responses during breath hold types, and this helps illustrate the variability in dive response. Panel A, the top one, shows a single dolphin performing a long, short, and no symbol breath hold. Here we can clearly see the difference between conditioned and unconditioned trials. For the long breath hold, the dolphin decreases its heart rate very rapidly, showing a strong anticipatory bradycardia. For the short breath, this dashed line right here, the drop is much smaller and more gradual in comparison to this very dramatic drop right here. The no symbol trial, or the red line, is very interesting because the dolphin did not receive a cue telling it whether the breath hold should be long or short. In this case, its heart rate patterns fall somewhere in the middle. It's not as a deep bradycardia as the long trials and not as shallow as the short. So that was very interesting. Panel B down here shows several examples of the no symbol breath holds. Sorry. Um, where was I? No symbol breath holds in another dolphin. And there is a lot of variability in the heart rate responses. This suggests that when the animals are not cued and don't know about the breath hold duration ahead of time, their autonomic response is less predictable. For 50 seconds, we can see it's the lowest one of them all, and the red line, 82 seconds, shows that it's not purely reflective. It shows that learned and autonomic control. These dolphins had less predictable bradycardia without the cues. And here is figure two as well, the examples of instantaneous heart rate responses from individual dolphins against time before and up to the 40 seconds during their breath holds. So for my discussions and conclusions, I did notice a lot of key findings. Overall, the study shows that bottlenose dolphins have conditioned control over their dive response. They can lower their heart rate more quickly and deeply when they know a long breath hold is coming. This demonstrates that the dive reflex is not purely autonomic. It can be influenced by learned behaviors and cognitive anticipation. And this connects back to the class topics by illustrating how the autonomic nervous system can be modulated through training and behavioral contexts. It also offers cool insights on how marine mammals optimize their oxygen management during diving. In comparison to us, that's very amazing. And I added that mammalian diving physiology fact down there. For critical analysis and future directions, there were some limitations to the study. First of all, there were only a small sample size. 3 dolphins were used. Second of all, the dolphins were trained, which means their responses may not fully represent wild animals. Finally, the study was done in a controlled lab environment, not in real diving conditions. So possible improvements for the future. Future studies could include more subjects, more than 3 dolphins, maybe triple or a lot more compared to trained versus untrained dolphins, or incorporate additional physiological measures such as blood oxygen saturation, blood flow changes. I think these different perspectives could really help us understand more in depth what happens with that bradycardia during long and short-term breath holds. Another potential direction would be studying whether wild dolphins exhibit the similar behavioral anticipatory bradycardia during natural dives, or not. I think that would be really interesting to see what huge difference or small difference there would be with these trained and untrained dolphins. This is my reference sheet for the article and the source that I used. Thank you for your time and patience. That's it for my presentation. Thank you for your time. Okay, bye.